Okay, let's talk about gas exchange. Now that we've talked about how gas enters in your mouth or your nose, enters through the nasal or oral cavity, to the nasal or the oral pharynx, down the laryngopharynx, past the vocal cords, into the trachea. It enters the lungs via the right and left bronchus, to the bronchi, the bronchioles, to eventually the terminal bronchioles. And then enters the respiratory zone where you get the respiratory bronchioles, the alveolar ducts leading to alveolar sacs, which secrete a surfactant to keep them from sticking together. And these number in about 300 million alveoli in your lungs. So once the air is essentially in the alveolar, we need to understand how we can extract the partial pressures of oxygen and CO2 from the air and actually get it to the working tissues. So over here, let's start with inspired air. So air coming in to the alveoli. We're going to have a pressure inspired, O2. This is going to be 160 millimeters of mercury. Remember from our last video, we talked about Dalton's law, and that once we looked at sea level, 760 millimeters of mercury, and the concentration or the percentage of oxygen in the atmosphere is 21%. 21% times 760 gives us 160 millimeters of mercury in our atmospheric conditions. Leads us to inspired air of CO2, going to be 0.3 millimeters of mercury. The inspired air of nitrogen, 594 millimeters of mercury. You might have noticed this isn't quite the same as the 600 millimeters of mercury that we had mentioned before, but we also want to introduce something else into the equation or the system, water. There's moisture in the air, and this accounts for approximately 5.7 millimeters of mercury. This is all entering into the alveoli. Let me just label that. And then air is leaving at the same time, and in our expired air, we get a little bit different. So we're going to have (116 millimeters of mercury). See, we've extracted 46 millimeters of mercury from the inspired air. And since we all know we're not plants, we expire carbon dioxide. We use oxygen via our cellular reactions, cellular respiration, and we produce CO2. And as we can see from we started out as 0.3 millimeters of mercury, we now have increased it to 27 millimeters of mercury. We expire 570 millimeters of nitrogen, so we've decreased that. And lastly, water is 47 millimeters of mercury. So we've changed quite a bit from what we started out when we inspired atmospherically and to what we've expired via that. So it's important to know, though, what we're trying to do is we're trying to get the O2 from the actual inspired air into the left ventricle to be ejected via the aorta and to all of our cells down here, working tissue. It's going to use oxygen. And it's going to produce CO2. This is nothing new. We've known that. So the thing is remember we had dead space before. And as we get to all of this gets mixed with our 150 milliliters of air that are already hanging out in our trachea and our bronchial tubes, we end up getting our pressure at the alveolar of O2 equals 100 millimeters of mercury. CO2, 40 millimeters of mercury. And then our alveolar pressure of H2O is 47. And lastly, equals 573. So all these numbers are a little bit different through the cycling. But let's say the O2 is coming in, it mixes, we have an alveolar concentration of 100 millimeters of mercury. This 100 millimeters of mercury is going to be the arterial, or small a, O2, and this is going to remain at 100 millimeters of mercury here. Our P alveolar, arterial CO2 is 40 millimeters of mercury. Arterial water, 47. And our arterial nitrogen equals 573. These are just more showing us what's happening in this. This is the part that's actually very helpful to us. We need to know what's going in because remember, cellular respiration uses oxygen and then produces CO2. So we have our arterial blood gas, our ABGs. At this point, this number, 100 millimeters of mercury of oxygen and 40 millimeters of mercury of CO2, are traveling to our cells. The cells are going to use the O2 and they're going to produce the CO2. And after cellular respiration occurs, we have an increase in CO2. This is the venous CO2. This is 46 millimeters of mercury. And our venous O2 now is 40 millimeters of mercury. So we've extracted 60 millimeters of mercury from the arterial blood gases into the venous blood gases. This doesn't mean much to you because we're talking about drops in pressure. We'll eventually have to convert this into something usable like milliliters of something we can quantify a little better. So these blood gases, these venous blood gases, are going to remain and enter into the right heart. Remember, the right heart is going to enter into the right atrium, right ventricle, and then it's going to leave via the pulmonary artery. Deoxygenated blood is going to leave via the pulmonary artery to enter into the lungs. And then this is essentially the pulmonary artery right here. So we have our deoxygenated blood full of CO2. The concentration is going to be similar to what we see here. So we have our venous O2 equals 40 millimeters of mercury. P venous CO2 equals 46 millimeters of mercury. Venous water, 47 millimeters of mercury. And lastly, our venous concentration of nitrogen is 573 millimeters of mercury. So this is going to enter in here and be exchanged at the alveoli, mixed with the dead space. And we're going to see a concentration or our expired air reflect that we have extracted essentially 46 millimeters of mercury from the inspired air. We have produced 46 millimeters of mercury mixed with dead space. We now have 27 millimeters of mercury. Our nitrogen remained fairly constant, but we have increased the concentration of water when we expire our air. So this is the gas exchange throughout our system, from inspired air to the alveoli to the capillary, through the arterial blood gas used by the cell, CO2 produced and CO2 removed out via expired air.